Good evening. You can hear me, right? Good evening. Well, welcome to the annual Forbes Lecture sponsored by the Grass Foundation. I'm Catherine Carr, the current president, and it's my pleasure to do a couple of tasks before we go on to introduce our speaker of the evening. The first is very agreeable, where we have provided an award for John Moore and Anne Stewart, where the Grass Foundation acknowledges their outstanding intellectual contributions and generous support of our fellows over the years. They've contributed immensely to the excellence of the Grass Foundation program. And Anne, if you would be so kind. So, as many of you know, every year the Grass Foundation sponsors a lecture from a world-renowned neuroscientist. And this year, our lecture is from Catherine Dulac, and she's going to be introduced by my fellow trustee, Vanessa Ruta from Rockefeller, in just a few moments. Before we go on to talk about Catherine and her many achievements, I would like to begin by talking about the Grass Foundation, because that's why we're here. We're here thanks to the generosity of Albert and Ellen Grass. Many of you will know the story, but it's pretty cool. They first met in 1935 in the Department of Physiology at Harvard Medical School and were married the year after. Albert was an engineer who spearheaded the design and fabrication of the first multi-channel EEG machine. Ellen received her that's right. So here's one of the first model EEG machines. Ellen received her AB and Masters at Radcliffe, and then as a PhD student was an early student of the electrophysiology of auditory cortex. And the two of them were very successful in their manufacture of the EEG machine and the later foundation of the Grass Instrument Company. And they very generously were able to endow the Grass Foundation. And one of their particular objects was to recognize the many achievements of Alexander Forbes, which is why this is called the Forbes Lecture. Alexander was one of the founders of modern neurophysiology, and credited with building and with his colleague Catherine Thatcher using the first vacuum tube amplifiers for neurophysiology experiments. The Forbes family has many well-known members, not the least is John Kerry, who's the senior senator from Massachusetts. Here you can see both Sir Adrian, Charles Sherrington, and Alexander Forbes in Ipswich. They done a number of important experiments, and it was in the 30s that Forbes first met Albert Grass, who joins his team at Harvard. But in addition to his presence as a neurophysiologist, Forbes was something of a Renaissance man. Here's a picture of him next to a plane that he flew. He was an accomplished aerial photographer and map maker. It's a distinguished service in World War II to help the US Navy identify airstrip sites. So clearly, clearly a wonderful individual. Now, with that introduction to Alexander Forbes and a brief mention of the Grass family, I'd like to finish up by mentioning the Grass Fellowship Program, which is our summer flagship program here at the MBL. Our Grass Fellows are here, many past Fellows are here, and we hope that they, along with the rest of you, will enjoy Catherine Dulac's lecture. So I'm now going to hand over to my colleague, Vanessa Ruta, to introduce Catherine.
It's really a great pleasure and an honor to be here today, actually to introduce Catherine as this year's Forbes Lecturer. Catherine is the Higgins Professor in the Department of Molecular and Cellular Biology at Harvard University and an investigator of the Howard Hughes Medical Institute. She actually started as really more of a developmental neurobiologist. She received her PhD from the University of Paris, where she studied mechanisms of neural crest development with Nicole Le Dourien. And then she moved to New York to carry out postdoctoral work in the laboratory of Richard Axel at Columbia University, where she developed novel genetic methods that really allowed her to identify the first mammalian receptors for pheromones, which are, of course, the chemicals that are given off by animals that regulate their social behaviors. So in her own lab, Catherine really has traced the pheromone signals into the brain, and over the years has begun to unravel the neurobiological rules that govern social interactions between animals, both how they fight and how they mate. And I would say in the spirit of Alexander Forbes, Catherine really is a true pioneer, always working at the technical forefront, but still driven by basic biological questions and insights. And I would say what makes her particularly inspiring to all of us who have an interest in the neurobiological basis of behavior and beyond is just her incredible breadth and scientific fearlessness. She's really tackled the fundamental questions of what really determines how animals interact with one another at many different levels in the most incisive ways, spanning an understanding of the molecular mechanisms of how social signals are detected, all the way through to shedding light on the intricate circuits that underlie appropriate male and female behavior. So for her seminal work, Catherine was made a member of the US National Academy of Science, a member of the French Academy of Science, a Knight of the French Legion of Honor, and of course the recipient of many awards, including the Lounsbury Award, the Parental Research Award, and the Edward Skolnick Prize in Neuroscience. So I think, as many of you may know, actually the Forbes Lecture spends a few weeks with the Grass Fellows, and I would say that they really are in for a great treat, and it's very fortunate that they get to spend a few weeks with her in the lab. I would say we're also very fortunate to be here tonight to actually hear her seminar, which is entitled The Neurobiology of Parenting Behavior. So before I ask Catherine to come up to stage and welcome her, I just want to actually remind you to maybe turn off your cell phones so there are no disruptions. And I would like to just say thank you very much, Catherine, for being here. We're really looking forward to your seminar.
Thanks, Vanessa, for the very kind introduction. Thank you, Catherine, as well. Thanks for the Grass Foundation for inviting me. It's really a very special honor to be here, to give the lecture today, to have time to meet the Grass Fellows and more generally to meet this wonderful community here. I'm actually stunned by how many colleagues and friends I met tonight and these last few days. I know why now there's nobody in Cambridge that I can meet. Everyone is here. So today I will tell you about the neural control of parenting behavior. But more generally, what I'm really interested in, as Vanessa mentioned, are the neural mechanisms that control social interactions. Why social interaction? Well, we humans are social animals. Our brain is literally geared towards social interaction. For example, as we can see today, we like to be with each other. We spend a lot of mental time thinking about who we like, who we hate. We don't do very well when we are isolated. Social interaction, love, hate is one of the most potent topics for art, literature. So social interaction are really, really, really important for us. But more generally, animals also need to interact with each other. Animals also don't do very well when they are isolated. Even the laboratory mouse, when isolated in a cage for too long, start to develop very abnormal pattern of behavior. And so for me, it has always been a very fascinating question to try to understand What is special about the neural control of social interaction? And by this, I mean, what are the type of signals that animals need to detect in order to engage in social interaction? What are the specific brain areas that are involved in controlling specific social interaction at specific times? And then more generally, how are these different behaviors interacting with each other? How are they modulated according to the animal developmental stage, social experience? And what we are trying to do in my lab is really to address this question very mechanistically, which means try to define the specific set of neurons in the brain that are involved in specific type of social interaction, specific type of social behavior, and try to understand how they interact with each other, how they are modulated. So the analysis of parenting behavior is actually relatively new in my lab. I started to work on the control of parenting maybe 5 or 8 years ago. And I found this to be a really interesting and very fascinating behavior. Parenting is unlike any other social behavior. It has many very unique features that are quite different from other social behavior. We take parenting as a given. You know, we all have been the beneficiary of parenting, and many people in the audience have themselves being parents. We think it's absolutely obvious that one needs to take care of the young and enable them to thrive. But if you really think about, there's nothing obvious about parenting. Parenting is a very asymmetrical behavior. It's one adult taking care, or more than one adult, taking care of a very helpless, screaming, dirty, needy thing. So, you know, obviously, we know it's important. There is all the social pressure. We know it's essential for the survival of the species. But an animal many mammals, for example, are parenting. And, you know, they don't care about the survival of the species. However, they will sacrifice themselves to take care of their young. So this is interesting because it means that this behavior is driven by some type of genetically pre-programmed neural circuit, neural drive that push animals to undergo parenting behavior. So in addition, unlike many other social behavior, this bond, this social behavior is long-lasting. It takes years, decades in human, maybe the entire life. It involves care, sacrifice. It also involves more than 2 individuals. Particularly in human, alloparenting is extremely important because it involves the parents, the extended family, the sister, the brothers, the aunt, the uncles, the grandparents, the friends, even the entire social structures are help to the parenting task. And obviously, parenting is essential both for the emotional and the physical well-being of individuals. So parenting is essential. But parenting doesn't always go very well. And in particular, there is a pathological aspect of parental-infant relationship. And in particular, postpartum depression is an illness that affects 20%, up to 20% of mothers and 10% of fathers in the US. This prevalence is extraordinarily high, and there is very little said and very little done about it. The symptoms are extreme worry, dread, feeling of failure, difficulty in bonding with child, and often thoughts, obsessive thoughts of harming a baby. And there is no action in postpartum depression, but certainly these obsessive thoughts are very debilitating and really impair the emotional bond between the parent and the child. More rarely, but even more severe, is postpartum psychosis that affect about 1 in 1,000 new mothers, in which here the symptoms are very severe and lead to the hospitalization of the patient, because these obsessive thoughts of harming a baby can actually lead to actual harm. Now, what I'm going to show you next is a quote from a patient with postpartum depression, which I think really exemplified this very poignant, this agony of this difficulty in parenting. Sometimes I get really down and feel like, man, I can't do this. No one talks about the low moments, the pressure you feel, the incredible letdown every time you hear the baby cry. I have broken down I don't know how many times. Or I get angry about the crying, then sad about angry, and then guilty. Like, why do I feel so sad when I have a beautiful baby? The emotions are insane. This quote, this testimony of a person with postpartum depression is nothing special among people with postpartum depression. What is unique is who made that quote. Serena Williams, who is symbolized to some extent women's strength. So postpartum depression can affect anyone, even someone who can be seen as one of the strongest women on earth. So try to understand the control of parenting and what can go wrong in the control of parenting is, I think, a very exciting task. But before I'm going to go really into the topic of parenting, I would like to spend a little bit of time explaining to you why I started to be interested in parenting. And I think this introduction, relatively long introduction, will really give you some essential context to understand some of the key findings that we've made related to parenting control. So in the first part, I will tell you about what brought us to parenting. In the second part, I will tell you about the control of parenting. And in the last part, I will tell you about some very intriguing phenomenon in the brain which lead to actually the negative control of parenting. So first, let me just present to you the control of social behavior in its larger context. So social behavior is initially initiated by the detection of specific stimuli. They could be olfactory stimuli, visual stimuli, somatosensory, auditory stimuli. These stimuli are detected by specific sensory organs, and then this information is then sent to the brain, processed by specific centers, and then according to the internal state of the animal, lead to specific social behavior, such as aggression, mating, or parenting. What is an intriguing characteristic of social behaviors is that they are usually quite different. The responses to social stimuli are usually very sexually dimorphic, which is a particular stimulus will elicit a particular behavior in males and a very different behavior in females. So it's an intriguing question to try to understand how different these brain areas involved in social behavior are between males and females. One really important feature of the control of social behavior is that it involves indeed the detection of specific social stimuli, and these social stimuli are usually extremely species specific. So this is an example of a bird, the American flicker, in which the male and the female differ by this black spot on the face of a male. And intriguingly, male and female can recognize each other as male and female exclusively through the detection of this black spot. So if you paint over this black spot here on the face of a male, then this animal will be identified as a female, which is the other male are going to try to mate with this male without a black spot because they think it's a female. And similarly, if you paint a black spot on the face of this female, now the other male, instead of attempting to mate with that female, will actually attack it because they think it's a male. So I have a black mustache, I'm a male. I don't have a black mustache, I'm a female. These visual stimulus uniquely identify a male from a female. Similarly, many mammals, particularly rodents, identify each other through chemical stimuli, and then humans use a combination of various stimuli, mainly auditory, visual stimuli, as well as some internal cognitive function within the brain. What is essential, again, to realize is that these stimuli are extremely species-specific, but the circuit that process these stimuli and drive social behavior are thought to be highly evolutionarily conserved. So Again, an essential feature of social behaviors is that the responses are quite different between males and females. And so this has led quite a number of scientists to try to think about how different is the male and the female brain. And so sex differences are very prevalent in the morphology of animals in nature. And it is very well known that sex hormones are the drivers of these important morphological changes. And so similarly, one might wonder how different is the inside of the brain, the circuit within the brain of males and females. And a relatively widely accepted version, and a bit of a simple version, but again, somehow accepted, is that the brain and the male and the female brain are quite different from each other, and probably structurally different. And when people started to think about it, it made a lot of sense that there could be structural differences that are similar to the structural morphological differences between males and females that is observed in many species. But how true is this? And when people actually started to look and identify those differences, morphological differences within the brain, the results were quite disappointing. There are differences. For example, one can see some brain areas, for example, here in the medial amygdala, an area involved in emotional processing. They are a little bit more dendritic spine, so these little spines here that are involved in synapses, so in the connectivity between neural cells in one side of the amygdala in female compared to males, but these are relatively minor differences. There are other areas, for example, this particular nucleus in the preoptic area that is known to be larger here in the guinea pig compared to the same area in female. Now, this area called SDN, or sexually dimorphic nucleus, has been recognized as being larger in males about 40 or 50 years ago. But to some extent it makes sense because this area is thought to be involved in the control of male copulatory behavior. So it would make sense that there are more neurons of that sort in males than in female. However, these differences do not exist in all species, even in species in which males have a very different mating pattern than females. So the search for these differences structural differences that might explain the differences in behavior have not gone very far in most mammals, most animals. And so one difficulty that neuroscientists have is to be able to relate structure and function. It's possible that the structure does not look very different, but the function is actually quite different between these different brains. And so the approach that we have taken is a very different one. It's a genetic approach that I will tell you about. And this genetic approach really led to extremely unexpected results. So most terrestrial mammals detect social cues through the detection of pheromones, chemical cues in the air. And these facial grin that you see here, where animals retract their lips, is a way for them to detect social scent and have this scent reach a particular set of olfactory neurons in a structure called the voronasal organ. These neurons then connect to very specialized brain areas, the accessory olfactory bulb, the medial amygdala, and then the hypothalamus, which is an area specialized in driving social behavior, such as mating, parenting, and aggressive behavior. And these neurons differ from neurons that convey the main sense of smell, which are located in the posterior recess of the nasal cavity, this main olfactory epithelium, that then connect to the main olfactory bulb, and then multiple areas of the brain that constitute the olfactory cortex. Now, as you can see, the voronasal system entirely bypasses cognitive areas in the brain. They go directly from these neurons to this pathway and areas involved in directing social behavior. So the idea is that there is no or very little cognitive processing of pheromones in contrast to the detection of odorants. So many years ago, we identified a particular gene that encodes an ion channel called TRPC2. The name doesn't really matter, but what you have to know is that this gene is absolutely essential for the function of neurons in the voronasal organ. Without this gene, these neurons are unable to detect stimuli and convey the information to the brain. And a similar gene exists expressed by the neurons in the main olfactory epithelium, but because it's different, if one make a mutant for this ion channel, then these animals are completely unable to detect pheromones, but they can smell odorants in a normal way. And when we observed the behavior of this mutant, we were very surprised. What we found was absolutely not what we expected. In brief, we found that male mutants for this channel, so male mice that could not detect pheromones, were unable to tell apart a male from a female. So they were able to mate perfectly normally, but if you put a female, they would mate with a female, and again, these are males. If you put another male, instead of attacking the male, they would mate with that male. And if you put a male and a female together, intruder, then the mutant would mate indiscriminately with the male intruder and the female intruder. So in other words, they were completely unable to discriminate a male from a female. So that was quite intriguing, because we expected that the voronasal organ would just be essential for mating, and it's not. It's essential for the discrimination between males and females. So then we were interested in observing the behavior of the mutant female. And here we had enormous difficulty, because basically there was absolutely no literature that indicate to us the type of behavioral assay or the type of observation on female behavior that we need to do our study. And so here I'm going to take a little parenthesis and mention that there are actually very few studies on females in biology in general. And this is a huge problem. So I like this quote from Sarah Hardy that says, compared with Darwin's exquisitely detailed observation of barnacle, coral reef, and orchid, even the emotional development of his own children, this consummate naturalist observation of women and other female primates were curiously cursory. Basically, most scientists don't find female of any interest, so focus on the behavior or the biology of males, and when they are possibly interested, then they find it too complicated. So 2 reasons to eliminate. But the problem is that at this point, any drug that you find on the market, whether it's a pain medication, it's a medication for cardiovascular disease or anything, has been tested mainly and studied mainly on male models, meaning all these drugs worked relatively well on men and very more poorly in women. And this has really led to a red flag raised by Francis Collins several years ago to try to achieve some type of balance. But in terms of animal behavior, there is very little done on female behavior. So it was embarrassing for me, frankly, because I always thought as a female PI, I would always study in parallel males and females, and it didn't happen. And when I was presenting the data on our male mutant, I always had somebody at the end of my talk saying, well, what about females? And it took me quite a while to convince a postdoc to look at the behavior of this mutant. And what we found was quite stunning and really, really interesting. So what you see here in gray is a mutant female for voronasal detection, what I call VNO-. And this female is put in presence of a wild-type male. All we want to know here is simply whether this female is sexually receptive to the male. But the behavior we observed was quite unique. Again, this is a female. And as you can see, she displays a male-like mounting behavior. Now, interestingly, not only this female mates like a male, but she also emits ultrasound like male. So a number of behaviors are male-like instead of female-like. Now, this could be a developmental abnormality. There's a miswiring in this mutant. So what we did is that we performed a surgical ablation of the VNO in that female, in an adult female. And what we found is that the phenotype was identical. So in other words, this is not a developmental anomaly. This is really due to the function of the voronasal organ in somehow repressing some type of male-like circuit. And when you remove it, either surgically or genetically, then these females behave like a male. Interestingly, also, all these mutants have normal level of steroids, which is a really important control. And so it seems to be the case then that the voronasal organ in normal female silences male typical behavior circuit in normal females. So here, I need to take a little parenthesis and remind or tell you how is the gender of the brain established. And the idea is that the gonads very early secrete specific steroids, estrogen and progesterone in female, and these lead to the feminization of both the body and the brain. And in males, the testes secrete testosterone, and this is essential for masculinizing the brain and the body. But what happened, what is really important is what happened very early. Indeed, this is in human. What you can see in blue are the level of testosterone prior birth. And you can see these two surges of testosterone in male embryos. So in human, it's perinatally. In mice, it's right after birth. But you can see in female, there's no surge of estrogen until puberty. But in male, there are these two peaks of testosterone very early in the newborn and the embryo. And those are thought to be essential to organize the male brain. And so females that do not have this surge of steroids develop female-specific circuit, and males that have this surge of testosterone develop these male-specific pathways. And these circuits are then silenced until puberty, when there is another surge of hormone, both in males and in females, and that leads to female-specific behavior and male-specific behavior. But there is a problem, which is that this model actually does not fit at all with the data we have obtained with our mutants. And so what we proposed several years ago is a very different model in which both the female and the male brain have both male and female pathways. And the key is that in each sex, there is a particular switch or control element that normally represses male-specific pathway in female and activates female-specific pathway. And we don't know whether this is the case in male. We hypothesize that something similar exists in the male brain, which is the male brain has both male and female pathway, and then normally the female-specific pathway is inhibited. In other words, the male and the female brain are literally the same, but the regulation of the pathways is different between males and females. And there is actually a lot of literature that suggests that this is the case. So all the old literature shows that in all species examined, normal animal behavior includes male and female features. So in other words, before the discovery of steroid hormones, when suddenly all the males behaving like males, all the females behave only like females, in this earlier time, Stone and Beach observed that males occasionally show female-specific behavior and female occasionally show male-specific behavior. Moreover, there are quite a number of animals, reptile, fish, fly, in which one animal can switch from the behavior of one sex to the other. And there's one particularly striking example discovered by David Cruz at the University of Austin, which is the example of the whip-tailed lizard. These species, the particular species that I show here, is parthogenetic, so they're only females. And the females reproduce by cloning. They basically lay an egg, and the egg is diploid, and it keeps going the species like this. But the ancestral species had males and females in which the behavior of these two sexes were very different. And in particular, the male has this particular copulatory behavior where it rolls around the body of the female in this donut-shaped behavior. Now, interestingly, in these parthogenetic species, A female can only lay an egg if another female adopts the male-like behavior, this donut-shaped behavior. So after a while, the he-female wants to lay an egg too. So now the she-female will adopt this male-like posture, etc., etc. And so these are females that are able to switch from male to female behavior, and they clearly have the neural substrate for both behavior. And similarly, in the human brain, there's been no categorical male-female differences, but a mosaic of traits that was found. And so the idea here, again, is that the male and the female brain at the structural level might be largely similar, but what will be essential to the control of sex-specific behavior is the modulation, the modulatory pathway of this particular behavior. And so this is what led us to parental behavior, because we were looking for a female-specific behavior and try to see whether the circuit for that female behavior would indeed exist in the male brain. And so parenting in mammals is mainly provided by maternal care. And it makes a lot of sense since the female, through gestation and nursing, invest an enormous amount of resources, and therefore it's her pressure, certainly, to make sure that her investment can thrive and develop as an adult. The behavior of male is quite different in many species. And Sarah Hardy, who was a graduate student in the psychology department in the 70s, made this extremely interesting discovery of infanticidal behavior in males. And she was observing this species of primate, the langurs in India, and what she found is that each time this male entered a social group, foreign social group, all the infant would disappear or be wounded. Now, the weakness of her study is that she actually never observed an attack. She just observed the result of the attack. But what she understood was that there was actually an evolutionary drive for this male to attack and kill infants. And that is that this male had only one way to be able to sire their own offspring, which is to kill the offspring of their rival. And then the female, who were normally taking care of the offspring of this rival, would now go back into oestrus and be receptive again, right? And so as it turns out, about half of the mammalian species have males that are infanticidal. And at first, this discovery of this hypothesis generated an uproar, because nobody would believe that primates, that in many cases are studied as a proxy to understand human behavior, could really have this absolutely awful behavior of killing infants. As it turns out, many primates do, and lions do, many rodents and many mammals display infanticidal behavior. Now, not all mammalian species display infanticidal behavior, and in fact, there seem to be some adaptation in some species against infanticide. So, for example, monogamy, in which male and female stay together, or dilution of paternity, in which the female mate with multiple males, and therefore no male can know which one is its own progeny, are thought to be adaptation against infanticide. Males are not the only sex that can be infanticidal. It is well known that in the wild, females that are stressed because there are too many predators or because there are not enough food also commit neglect or infanticidal behavior. And certainly, it is very striking that human postpartum illnesses have as a very important risk factor stress. So, we were very intrigued by this female-specific behavior. In mice, virgin females are robustly parental, so even if they never had their own pups, they will take care of the pups of other females. They will build a nest and retrieve them to the nest. In contrast, virgin males are robustly infanticidal. Within a few minutes, they will attack pups and kill them. So very clear secretomorphic behavior. But if we take voronasal mutant, again, this system that seemed to regulate the sex specificity of female behavior, now we found that they no longer attack infants and in fact display genuine parental care. So that's, complete our model, if you wish, that the male brain has this female-specific parenting circuit that is normally repressed by the voronasal organ. Now, you might wonder, why would the male and the female brain keep the behavior pathways of the other sex? Well, it turns out that maybe it's useful sometimes. Maybe it provides some flexibility in behavior. And indeed, there's something quite extraordinary that happens to the behavior of male mice after they mate with a female. In fact, fathers switch their behavior from infanticidal to paternal behavior. And this switch does not occur immediately. It occurs after 3 weeks after they have mated with a female. And 3 weeks correspond to the gestation time in mice. So 3 weeks after mating with a female, any pup around could be their own. They are actually unable to distinguish their own pups from that of others. They can't tell them apart, but the timing provides them a way to say, these might be my pups, I better not kill them. Before that, any pup around are unlikely to be their own, and they kill them. So the video I'm going to show you here is a male that 3 weeks earlier was infanticidal, certified infanticidal male. We had that male mate with a female, and as you can see, 3 weeks later, this male is dutifully retrieving his pups, has built a nest, and, he behaved like a perfect father. And in fact, even, make sure that he didn't leave anyone behind. And then goes back to the nest, and then we crouch around the pups. So this is very striking change in behavior, and clearly indicates how the male brain contains perfectly functional parental circuit. No excuse, guys. Now, this is interesting at the individual level, but it's even more interesting at the evolutionary level, because, we always think about parental behavior as the behavior of females, but actually there are many species that are parenting, not only mammals, birds, reptiles, here frogs, fish, and even insects. In these species, the female is the only one providing parental care, but very similar species show biparental care or even maternal, male uniparental care. So, these bugs, they look very much similar to each other. These frogs here, these poison frogs that are studied by my former colleague, Lauren O'Connell, you can see on the top here of this female frog, or this male frog here, and here a tadpole. So this male here is protecting this tadpole and will carry the tadpole to a source of water. These species are very similar to each other genetically. So it's very unlikely that this particular species has only parenting circuit in the female brain and then here only in the male brain. All of these species have neurons that might drive parenting behavior, but in these species, these are activated only in females, here only in males, and here both in males and females. And this is true for many of these animal species. So basically, the idea of having male and female brain to be very similar to each other, but very regulatory mechanism provides a lot of flexibility at the individual level within a species, but also probably at the level of different species throughout evolution. And so in mice, mice have this little harem. There is a dominant male and a group of females, and the female have communal nests. So the female bring and raise all these pups together. And the dominant male is continuously mating with the female. So this male is continuously paternal, in contrast to the subordinate males that are infanticidal. And this male will only be able to mate the female if they kick out this dominant male, and then they will kill the pups and then mate with the female and become paternal in turn. So at the neural level, this is very interesting because it shows that there are two types of pathways. The pathway used by virgin males to drive infanticidal behavior, and the one used by fathers and females that drive parental behavior. And we are just really interested in understanding what are these neurons that control either parenting and infanticidal behavior. So to identify neurons involved in parenting, we used a molecular trick, which is that neurons that fire during a particular behavior also change their gene expression. And in particular, they start to upregulate a particular gene called CFOS. And so if we let a female or a male interact with pups for about half an hour, and then we sacrifice the animal and we look for areas that are highly enriched in CFOS expression, it will tell us the areas involved in that particular behavior. So if we do this experiment in parental males and females and compare the brain to that of infanticidal males, it was very easy for us to identify a particular area of the brain called the medial preoptic area, in which there was a lot of CFOS expression, meaning a lot of neuronal activity, when this animal interacted with pups compared to infanticidal males. Now, this is not surprising. The medial preoptic area has been known to be involved in maternal behavior for decades. Moreover, these areas of the brain not only is involved in parenting, but also in thermoregulation, sexual behavior, aggression, thirst, and feeding. So in other words, there are a lot of neurons here that do a lot of things. And the key for us was to be able to identify specifically the neurons that express CFOS that were involved in parental behavior. And by different assay, candidate gene, we found one particular gene that encodes this peptide called galanin that was very nicely co-expressed with neurons expressing CFOS during parenting behavior. So we think that this peptide galanin is a marker of neurons that are involved in parenting behavior. And I should say, we do not know whether galanin itself is involved in parenting itself. It's just a gene expressed by neurons involved in parenting. And I should say these neurons also express an inhibitory neurotransmitter called GABA. And it's possible that GABA or galanin or both of them are the ones that fulfill their function. So in a subsequent set of experiments, we really tried to see whether these neurons indeed were involved in parenting. And for this, we did two sets of experiments. One is using tools that I won't describe, but are genetic tools that enable to specifically either kill the neurons or activate them. In the first set of experiment, we ablate this galanin neuron in the medial preoptic area. And when we tested females that were depleted in MPOAG galanin neurons, we found they were no longer maternal. And when we did the experiment in fathers, we also found that they were no longer paternal. So the ablation of these cells abolishes parental behavior. And even in virgin female, we found that now these female without galanin neurons in the preoptic area were infanticidal. In a reverse experiment, we now use new tools of neuroscience called optogenetic tools that enable to specifically activate by flashes of light a genetically identified neuronal population. And when we did this experiment in virgin males that are normally infanticidal, we found that now these males were no longer infanticidal and display grooming behavior of the pups instead. And so what that tells us is that these neurons, these galanin neurons, are both necessary and sufficient for the control of parental behavior, both in males and in females. And I should say these galanin neurons play exactly the same role in males and females. They are present in exactly the same number. So there's no difference between males and females. There's a difference only when the normal expression of the behavior is displayed. So it's interesting to discover a population that has a particular function like this, and clearly it's intriguing to think about other species. Are other species also relying on these MPOAG galanin neuron to drive parental behavior? And I don't know the answer to this. As I mentioned in the introduction, these central circuits for behavior are usually highly conserved. So it's quite likely that from the frog to primates, maybe even human, the same set of neurons play a similar function. This is the case for neurons that control sleep, feeding behavior, reproduction. Many neurons that are involved in homeostasis or social function are conserved across species. And interestingly, very recently, Lauren O'Connor at Stanford and Andy Bass at Cornell found that galanin expression was upregulated in species of poison frogs or species of middleman fish that are parental. So it's possible that these animal at least, also rely on the function of these galanin neurons. Now I'm going to show you how extraordinarily specific the activity of these neurons is. So what we've done here is an experiment called photometry, which is we are recording the activity of a population of neurons by virtue of the expression of a calcium indicator, a fluorescent indicator that will shine light when the neurons are activated. So what you will see here is a trace, a fluorescent trace. When the trace goes up, it means there is a lot of fluorescent signal, meaning there's a lot of calcium released, meaning these neurons are active. And when you can do this in a behaving animal, so this is a female, and you'll see the female has an optic probe that sticks out of her head, and she will approach these pups here and then retrieve the pups to the nest. And as she does this, you will be able to see the increase in calcium in neuronal activity. So here, the female approaches the pups, and you see an increase in neuronal activity, and then it will tampen down a little bit, and then she will bring the pup to the nest. And as she does that, the calcium signal will. So now she's starting to groom the pups, and as you can see now, the neural activity increases dramatically. In the next video, something very similar happened. You can see this animal, this female here, licking something, and as you can see, there doesn't seem to be any neural activity. And the female, in a few seconds, will retrieve to the nest, and again, absolutely nothing is happening. So what's going on? Well, this is actually not a pup, it's a fish cracker. So to an observer, it looks like this animal is doing exactly the same thing as parenting, grooming, licking, retrieving to the nest, but in the brain of this mouse, this is feeding. And so these galanin neurons have nothing to do with feeding. They are just only interested in parenting. And so we've done many controls like this, and the only behavior in which these neurons are activated is parenting. So now we would like to know how these neurons work. Parenting seems like relatively simple behavior, but these neurons need to receive complex input about the presence of pup, the internal state of the animal, and then the behavior that are engaged in parenting are actually many. The animal needs to build a nest, sniff the pup, lick, retrieve, crouch, and nurse. And in addition to this behavior, there are also a lot of aspects of the brain function that are different in parents. The motivation to interact with infants. A virgin female will approach pups, but the mom or dad will approach a pup very readily, way faster, and will engage into even go over hurdle to retrieve their infant. Hormonal levels are different in parents. The meaning of sensory stimuli is very different. And also, animals that are parenting are no longer interested in adult social interaction. So, before having infants, you go to the pub and have fun with your buddies. Now you have children, you stay home and watch TV with your children and forget the bar. So how does that work? Well, what we've done in the first step is first try to see what are the brain areas connected to these galanin neurons. In other words, what are the projections of these galanin neurons? What are the target area of these neurons? And for this, we use viruses that enable us to see both the axon as well as the synapses. So what are the synaptic partners of these neurons? And what we found was way more than what we had anticipated. We found about 20 areas that are the target of these galanin neurons. Among those, we can find the function for some of them, maybe in motivation or motor control, social behavior, or hormonal modulation, just by the name of those brain areas. But, clearly, it's very complicated. So we tested a hypothesis that maybe these areas were reached by different population of galanin neurons. In other words, there might be different pools, different population of galanin neurons, each one directed to a different target. If that was the case, that would make our analysis way simpler. And indeed, this is what we found, which is that if we injected dyes of different colors in different target areas and then looked then at the level of the preoptic area, we found that there were never two colors in the same cell, suggesting that these neurons do not bifurcate. In other words, the hypothesis that indeed these neurons, each neuron contacted only one target seemed to be true. And this is important for us because then we can study one target at a time and try to see what it does. And what I'm going to tell you very briefly is our work on 3 of these brain areas. They are indicated only by acronyms. I'm sorry about it. This one, PAG, stands for pericortical gray. This is an area that is usually involved in the motor control of social behavior or instinctive behavior. The VTA, or ventral tegmental area, this is an area that is involved in reward and maybe motivational aspect of behavior. And the medial amygdala, which is involved in a number of social behaviors. And so what we did is to manipulate specifically the neurons that project to one or the other of these areas. And what you'll see is the results are really quite interesting. So we start by the pericortical gray. And the idea here is to activate the terminal of these galanin neurons to the pericortical gray and to see whether this activation changes the interaction of a male or a female with pups. And when we do the experiment, we found that indeed activation of this terminal increased parenting both in males and in females. I'm going to show you very few data, but this is one of them. So here's the laser off. These female have some amount of parenting, but not much. And when we activate this terminal, we see an increase in parenting. This is the experiment done in males, and the males are normally infanticide, so they are not parental. Now, when we activate them, we found that the very few males are infanticidal and none they are parental. So we found no change in other social interaction, no change in locomotion. And so what we concluded from this is that this particular connection from galanin neurons to the pericortical gray drive the motor execution of parenting. Then we looked at the VTA, the ventral tegmental area, an area that is involved in reward and motivation. We did exactly the same experiment as what I just described, and we found absolutely no effect on parenting, nothing. Okay. Then we thought about the following. We remembered how much effort parents go through to reach their infant. And this is exemplified here in this video, where this female has her pups that are in this tall beaker here. She just realized that and tried to reach them. And obviously she cannot through the glass. And so you'll see how much effort she's going to go through in order to reach her pups. Okay, she realized maybe she can reach them here. Okay, and then some acrobatic. Yes. And here she goes. And in the next few minutes, she will retrieve all her pups. So, this is a bit of a complicated test. What we did instead is just having a barrier between the female and the pups. And when we do the experiment, indeed, when we activate the VTA, we find that the female crossed the barrier with a higher probability. Now, interestingly, when we do the experiment in males, we also find that the males also cross the barrier with a higher frequency. But these are virgin males. And when they reach the pups, they kill the pups, right? So you have to remember that here we're activating only one target of these galanin neurons, the one that drives motivation to reach the pup. We are not activating the terminal to the pericortical gray that drives the parenting itself. So this animal will have increased drive to reach the pups, but they are not parenting, and therefore they kill the pups. And I think it's a very good example of how separate the control of the various aspects of parenting is. And then finally, we did the same experiment to the projection to the medial amygdala. And here we found no effect on parenting, no effect on motivation. But now, when we have a male and a female interacting to each other, we found that there is a very strong decrease in social interaction. And so what that tells us is that the system received quite a lot of input. I didn't have time to show you those data. But the important part of the circuit is really the existence of this separate population of galanin neurons that each one perform a particular role. The motor aspect of parenting, the motivation aspect, the social interaction aspect, the neuromodulation that I didn't have to tell you about, and then also another aspect which is inhibiting infanticidal behavior. So this is a very unique mode of organization in this different population doing different things. So let's go back to some aspect that I mentioned to you, observed in human, which is this very strong increase in psychiatric hospitalization right after childhood. And this is thought to be, so this is women with postpartum psychosis, and this is due in part to what is described as a hormonal chaos at birth, when hormones that have steadily raised during pregnancy suddenly drop. And this has a tremendous impact on the mother's brain and can lead to some psychiatric illnesses. And so one very intriguing part of the literature on parenting is that people have described brain areas that increase parenting and also some brain areas that decrease parenting, suggesting that there might be both a positive and a negative control of parenting. As I mentioned to you, infanticide actually in many animal species is a normal physiologically relevant behavior. Male infanticide to have access to the female, and female infanticide in case of a dire condition, environmental condition. And so we turn now to try to understand how is this infanticidal behavior controlled. And using a very similar approach as what I had described to you with parenting, we were able to identify a particular set of neurons in this area called the periphonical area, also in the hypothalamus, that are exclusively activated during infanticidal behavior. And it took us quite a while to identify the nature of these cells, but we finally found a marker, this peptide called urocortin 3. It's a peptide, and interestingly, it's this peptide and neurons are part of the stress circuit. And as I mentioned, stress is consistently associated with neglect and infant aggression. Now, we wonder how specific is the control of this pup-directed aggression. Maybe the neurons that control this aggression to pups are the same that control other forms of aggression. And that doesn't seem to be the case. What you see here in green are neurons involved in adult male aggression, adult to adult. And they are different from the cells expressing urocortin 3. We tested any kind of aggression we could think of. Maternal aggression, defensive response to predators, anything. And what we found is that these urocortin cells are exclusively activated during infanticidal behavior. It's kind of weird. It seems to suggest that they are indeed a dedicated neural system for aggressive behavior. Now, is that true? So when we silence these neurons, we found that virgin males are no longer infanticidal. And interestingly, the effect is long-lasting. Way after we silence these neurons, the males are no longer infanticidal. And similarly, when we do the experiment in females, I'm going to show you a video here. Here we activate neurons in female. And what we found is that they now show sign of aggression. And the one telltale of aggression is tail rattling. You see this female approaching a pup here. Now we're activating these neurons, and you see this tail rattling, which precedes attack. So these neurons are here again, both necessary and sufficient to trigger aggressive behavior. I'm running a bit out of time, but I would like to tell you how these neurons work in exactly similar way as what we have done with parenting. So these urocortin cells have many projections, and we just looked at 3 of them. They were the most abundant projection, and also these projections are known maybe to be involved in aggressive behavior. But as you'll see, the activation of these different projections will lead to very different type of behavior. So, for example, we activated the projection of these neurons to an area called the VMH, or ventral medial hypothalamus. This is an area that is thought to be involved in male-male aggression, adult aggression. And we thought You know, this is the way it works. These urocortin cells activate VMH neurons, and therefore that trigger aggression. And we were wrong. We were completely wrong. So here's what happened. Here's a female and a pup. And what happened is when you activate the neurons, the female hover around the pups. So instead of reaching the pup and bringing it back, she just can't seem to be able to touch it. So it seems like this may lead to pup neglect. Then in a second experiment, we now activate another set of projection to this area called the lateral septum, which is an area that is known to be involved in anxiety and stress. And so you have to pay attention here because the behavior is very quick. Here's the female here, and here's the pup. When the female will reach the pup, then we will activate this projection, and you'll see something very brief but very obvious will happen. Here's the female approaching the pup. Whoops. This is very brief. These females seem to be fleeing, escaping the pup, and she will never get back to the pup, ever. So these pups would be just totally abandoned. And then finally, we activate this other area, this AHI area. And I'm not going to show it to you because it's a bit gruesome. But in this case, the female grabbed the pups by the belly and just carry it around like this, exactly like an animal that would commit infanticide. So what I've shown you is now this brain area here that receive input. I didn't have time to describe it to you, but various input related to infant cues, stress, physiological state, and these neurons reach a number of other brain areas that drive various aspects of neglect or aggression towards infant. Interestingly, these neurons are actually directly or indirectly connected to the neurons I mentioned before, these galanin neurons. So what we have here is these two pathways that drive opposite behavior, that are connected to each other through inhibitory connection. So when one pathway is activated, it inhibits the other one, etc. And each one has this module that drive very specific aspect of parenting or very specific aspect of neglect or aggression towards infant. And so what these provide is a vast range of flexible behavior using these hardwired circuits that are both present in the male and the female brain. So we started our analysis by thinking about male behavior, male brain, female brain. And what we found is that these two opposing circuits are both present in the male and the female brain. And therefore, at the individual level, this provides a lot of flexibility. No individual will be identical to the other one. Some will be more maternal, less parental. So in the animal kingdom, each species will have variability around a particular theme. And this flexibility also is probably quite noticeable among various species. Some species have only the male taking care of infant or only the female or both of them, and they probably both rely on identical circuits that are simply modulated differently. And then finally, what do we learn or what can we get out of this related to the healthy or diseased brain? And at this point, I don't know. It's always very difficult to think about how a discovery, particularly in neuroscience, can impact directly mental health. But the fact that we know now some of the key neurons that drive either parental behavior, normal parental behavior, or that negatively regulate parental behavior could be a source of knowledge of modulators that can impact parental behavior and that might possibly help patients. And so I will finish by acknowledging the people in the lab who've done the work. The initial work on parental behavior started by the work of Herbert Wu, who is now a postdoc at Columbia, was a graduate student. The follow-up on parental behavior was done by Johnny Cole, who is now a faculty at the Crick Institute in the UK. The circuit driving infanticide behavior was done by Anita Autry, who is now studying her lab at Einstein College of Medicine. And the older work on sex-specific behavior was done by Talim Kimchi now at the Weizmann Institute. And we have wonderful collaborator, Nao Shida at Harvard, his postdoc, Benedicte Babayan, who is now an editor on Neuron, Venki Murthy and his postdoc, Vic Kapoor, and Lichun Lu at Stanford. Thank you very much.
Catherine, thank you so much for a wonderful presentation. We have some time for questions if people would like to ask. You should put their hands up. We've got 2 microphones. And then I'd also like to remind you that we will have a reception following the talk in Swope.
Catherine,
Yeah.
am I on? Yes.
Microphone wasn't on.
How about that? No? I'll shout. I'm wondering if you would be willing to speculate a little bit about the postpartum depression in humans, which might suggest a conflict between the behavior that is being invoked that seems to maybe conflict with either societal norms, or is there a neurobiological basis for that conflict that's driving the sense of depression in those individuals? And might that suggest circuitry that would be, driving that sense of conflict and depression in those individuals?
Yeah, so, first of all, I knew nothing about postpartum depression. And when I saw the numbers, 20%, I thought, I don't know any mental illness that has such a high prevalence and that seem to occupy so few people. That seems unbelievable. Now, if you think about one risk factor of postpartum depression is stress. Stress, social isolation, and these, to some extent, in animal, stress and social isolation, females are just gonna, neglect their infant as well. So there might be some evolutionary drive here that reduce parenting in these circumstances. Another one is simply the sensitivity to these enormous hormonal fluctuations. So the sensitivity to hormone changes in menstrual cycles, for example, is also a risk factor for what's for this steep decline of hormone after birth. I don't know. Interestingly, actually, postpartum depression also exists in dads at a lower level, but also exists. And here, we know that there are hormonal changes in mothers. Obviously, we know because pregnancy, etc., gestation require changes in hormone. We know nothing about what's happening in fathers. And I'm not talking only in human, in animals as well. These male that mates and then 3 weeks later becomes parental, something has to happen in its brain. Maybe it's hormonal, maybe it's a circuit level, we don't know. Nobody knows anything about it. So there are all these changes. I don't know if this is answering your question, but...
Maybe we can talk about that.
Okay. Yeah.
So, Catherine, given that humans don't have a VNO, how much of this...
Say again?
Do humans have VNO? And if they don't, how much of these lessons are applicable to human...
Yeah, so this is exactly why I mentioned at the beginning that it's important to consider that every species has its own set of stimuli. And in mouse, it's the VNO. In birds, it could be visual or auditory, and humans have their own set of sensory stimuli. But the core circuit, the hypothalamic circuit, are very similar, irrespective of the sensory trigger of those. It makes sense that the sensory signals are different because every species has its own niche and has its own type of rules for social interaction. but the core circuits are very similar. You know, parenting behavior across animal, it looks very, very much the same. The drive is the same. If you look at mating, aggression, feeding, sleep, thirst, you know, all these survival behavior are likely to rely on very similar circuits. What will differ is the way they are modulated, obviously.
My question also deals with the VNO in mice. Clearly, you started your lecture, beautiful lecture, by indicating that that is a dominant regulator as an organ in parenting and other behaviors. So, what is known about specific pheromones? So it must be sensing specific pheromones. Is something known about specific ones where you can even play with neurobiology to block activities without taking out the entire organ?
Yeah, so this is a good question. If I had one more hour, I would have tell you the story about those particular pheromones. So we actually know what they are. It's a very interesting story. And yeah, but, you know, to get back to Rafi's question, you know, there are many primates that display infanticidal behavior, very robust infanticidal behavior, and they have no VNO. The triggers are different. Could they be auditory signal or olfactory? We don't know. So again, you know, the stimulus itself is not that critical. Yeah. So you're talking now about the children's side. How?
I'm looking at, you know, those kind of critical years for a female, starting at 10 and up, and how trauma might impact, you know, brain development over time.
So it's a very interesting question, because this has been shown in many animal species, actually in primates and also in mice and in rats. Infant neglect lead to young females that become neglectful mothers. If they are neglectful mothers, their own daughter will also, right? So there is a sort of cultural transmission of neglect. And, you know, there's a whole field of neuroscience that look at how stress affects both the behavior of the future adult, but also all its endocrine function, cognition, etc.
Catherine, I have a question here. So this is really beautiful work showing all of these different circuits and how they're responsible for different aspects of parenting behavior. I think it's really important to dissect all of that. We started, and many of us have kind of grown up with the sense or heard through most of our scientific lives, the sense that gonadal steroids are important for these kinds of behaviors. And so I'm not sure that we've reconciled from what you've done here, or what you've shown us here tonight, what is the role of testosterone and estrogen in modulating these behaviors in males and females? Can you give us your perspective on that?
Yes. So, you know, when we publish our work showing that these females, mutant female behave like males, I received a lot of hate email from neuroendocrinologists. They hated it. That was like 12, 15 years ago. Then now they keep inviting me to give their big lectures because they recognize there's something there. You know, basically, hormones do exactly what they were proposed to do in terms of modulating, regulating the function of neurons. What they might not do is to change the structure of the developing brain in males and females. And it's very interesting sociologically, because when steroid hormones were discovered, that was also the time when people were discovering growth hormones, growth factors. And so, you know, if you put growth factors in a dish and you see, you know, these neurons that send their axon and dendrites, etc. And so when steroid hormones were discovered, and they have such an enormous effect on animal behavior. If you castrate a male, it does not display male-specific behavior. If you inject testosterone in a young female, she will display male-like behavior. You know, all of these suggested that what the steroid hormone were doing is indeed, in male, generate establish this particular architecture, which is male-specific, and in female, direct a female-specific architecture. And I don't think that's the case. I think the hardwiring is the same, but what is different, however, is the sensitivity of those neurons to particular hormones. And so, again, if I had one more hour, and I will actually talk about this at the lecture on neuroscience on Wednesday, we now know what are the receptors expressed by these cells. And the neurons that control parenting behavior are full of steroid hormones. Androgen receptor, estrogen receptor, progesterone receptor, prolactin, you name it, and many, many peptides. So the key here is to think that the circuit for many behaviors exist in the brain, and they are triggered by particular input, neural input, and regulated by a particular physiological context that is provided by steroid hormones. So yes, hormones are very important, maybe not necessarily in exactly the same way mechanistically as was anticipated. It's not structural, it's regulatory.
Last question.
Time for one more question.
So thank you for the really wonderful talk. I was just wondering if you had any idea about any mechanisms of experiential plasticity in these circuits. For example, you mentioned the virgin males. Once they become parents, they switch from infanticide to actually taking care of the pups, and sort of what might regulate these changes in the circuits from experience.
I don't know, and we have certainly tried to understand that. What I can tell you is that now we have a better sense of what's going on at the circuit level, which is that, but we don't know how it's regulated. We know that virgin females have a core circuit that drives parental behavior, that parents have certainly additional neurons that participate in parenting, and interestingly, these neurons express the oxytocin receptor. So, right at birth, there is a peak of a release of oxytocin, and now you have this additional neuron that will lead to the way more efficient parenting of mom and dad compared to the virgin female. And infanticidal male just use different set of neurons. What makes the switch? We don't know. It's a fabulous question, and I hope we'll find the answer. Thank you.
And we'd like you to join us for the reception in Swope.
Thank you. I was worried it was too long.